Hello all, this is Vikash Google. I am an MTech student at Department of Biotechnology, IIT Madras, and I'll be presenting on the heroes versus villains, the role play of media components and process conditions in the oxidative balance of recombinant Chinese hamster ovarian cell culturing. So basically, there's an increased focus on biotherapeutics in the current decade, and Chinese hamster ovarian cells are the platform host cells for this production. The objective of this process development would be to achieve a high productivity and high product quality ensured. But the challenge would be the intracellular ROS generation and the ER stress due to the recombinant protein production. ER stress is nothing but the stress caused in endoplasmic reticulum due when the ER is forced to operate at its maximum or over the maximum its capacity. So we'll be speaking about the pro-oxidants, which are the villains in this case. The first thing would be cysteine. Cysteine is a semi-essential amino acid which can be produced endogenously, but still exogenous supplementation also required. And it can oxidize from cysteine to cysteine in the presence of air, iron, and other media components. And as we all know, it's a principal component in glutathione synthesis. So when the concentration is more than 2.5 millimolar and at a very low cell density, initial cell density, this leads to a reduced cell growth and intracellular ROS generation. And a differential experimental analysis by bulk RNA-seq showed that there is an increase in AMPK pathway, which means there is an inefficient energy metabolism happening, and there is a P21 mediated pathway which arrests the cell cycle and leads to apoptosis. There is an increased DNA damage and unfold protein pathway, which is indicative of the ER stress. And also there were genes associated with the redox homeostasis and stress protection has been activated. Whereas when it is in depleted, which is less than 15% of the actual present in the chemically defined medias, they found that there is also an oxidative stress produced, ER stress, and reduced viability. And this is because the cell tries to produce cysteine by degrading the glutathione. And you can see the red ones indicate the upregulation and green ones indicates downregulation. The glutathione is being downregulated due to the upregulation of the glutathione breakdown enzyme. And also we can see the accumulation of methionine and serine because the enzyme which is going to produce cysteine from these two amino acids has been downregulated. And also cysteine is the starter material for other antioxidants like hypothyroid and thyroid, which is being reduced in this case. So the net effect is inefficient antioxidant system and oxidative stress. So they also observed that the mitochondrial SOD has been upregulated, which is indicative of the mitochondrial dysfunctioning, which leads to ER stress, and subsequently it activates the DNA damage inducible gene 3, which activates the pro and apoptotic protein BCL, which leads to apoptosis. So the net effect is reduced viable cell density and viability, and also apoptosis. And the summary is that glutathione depletion has been increased and taurine biosynthesis has been reduced. There is an increased oxidative stress and mitochondrial dysfunction. So, but also when it is depleted state, the aglycosylated species of monoclonal antibody has been increased due to the reduced activity of the galactosyl transferases, and it also induces amino acid response and ER stress, which leads to increase in expression of phosphoenol pyruvate carboxylase, which converts oxaloacetate to phosphoenol pyruvate, and a reduced expression of pyruvate carboxylase, which converts pyruvate to oxaloacetate. So this leads to making the oxaloacetate unavailable for the TCA cycle, and the cell relies on anaplerotic pathways like glutamine lyase, which leads to increased ammonia synthesis. And this tries to replenish the alpha ketoglutarate to the TCA cycle. So basically the mitochondrial dysfunction is being observed, and in order to restore the NADH equilibrium, the cell opts to produce lactate, which leads to the increased pH. And also due to ROS, the glycerophospholipids present in the membrane is being degraded to free fatty acids, which is being converted into triglyceride. But this triglyceride is being stored as a lipid droplet rather than using it in beta oxidation due to the loss of key carrier proteins. So net effect is the reduced productivity of the protein. So DO is one of the important parameter in cell culturing. So when it is at 20% DO, lactate accumulation being observed, and there is a reduced consumption of amino acids like serine, valine, threonine, and proline and glycine. They quoted that this is because of the low productivity, but this trend is not seen in other amino acids as well, which indicates that this is not the reality, it's something different. And from our previous slide, we can see that serine is a principal component in cysteine synthesis, and which is again gets into GSH. So maybe that could be a reason why serine is not being consumed. And when at 100% and 175% DO, the viable density and viability is being decreased due to the production of hydrogen peroxide. And also there is no protein production at in this case. Probably the cell is trying to shut down the protein production and cell division in order to reduce the ROS generation and the energy requirement for these things. Thereby it can survive. And also there is an increased glucose consumption and lactate accumulation. And as we saw before, the cell opts for lactate production to increase the NADH equilibrium, and this is being complemented by the decrease in specific oxygen uptake rate. And also they found that the specific oxygen uptake rate with respect to the ATP synthesis being reduced in cases which indicates the poor OX-phos pathway and anaerobic metabolism. And this with respect to the sparging rate, they have done an in silico modeling, which is a genome scale modeling, and they proved that pyruvate being converted into a lactate and rather than converting into oxaloacetate. And as we discussed, it is for meeting the NADH requirements. The oxalate acetate is being replenished by asparagine breakdown. So the amino acid uptake is being increased in this case. So it tries to replenish the alpha ketoglutarate for the TCA cycle. And also at hard sparging, the hydrogen peroxide production is being significantly increased by 2.2 folds. And most of the amino acid metabolism being directed to hydrogen peroxide producing reactions, example like tyrosine oxidase and glycine oxidase. And there are also reactions to produce NADH and NADPH to replenish the energy requirements, like the aspartate dehydrogenase. And there are few reactions which produce hydrogen peroxide and NADH parallelly, like the L-phenylalanine carboxylase. And GS has been supplied from the reaction of D-lactaldehyde NAD plus oxidoreductase, which produces GSH and NADH and NADPH parallelly. So it's basically a collaborative effort of the cell to for the redox defense system, and the ROS generated in this case has been used as a signaling molecule to overcome the oxidative stress. So we know lyite is one of the component which induce ROS. They have observed that compared to a large scale production, in small scale supernatant, there is a 5 to 8% increase in acidic variance. The acidic variance is basically the negatively charged MAB molecules being produced. And they found that 58% increase in acidic variance in days 7 of the culturing, when the protein is being put in a formulation buffer containing riboflavin and tyrosine. And riboflavin produces hydrogen peroxide and superoxides. And when a quencher is being added, this thing has been reduced, which indicates that singlet oxygen is being contributes to the acidic variant production. And coming to the antioxidants, basically it is an alkaloid which has an antioxidant property, and it actually increases specific productivity, and it also maintains the aglycosylated species toward the process, indicating that it conserves the galactosyl transferase activity. And also methionine sulfoxanine is a selective inhibitor of glutamine synthetase, which is a marker in this CHO cells. And they found that this MSX can also inhibit a GCL enzyme, which is involved in GSH synthesis. So since the glutathione is being reduced, the ER stress induced actually upregulates the glutathione synthesis, which ultimately leads to the specific productivity of the cell. Also catechin, which is an alkaloid, it also being when supplied at a particular concentration, it can reduce ROS generation and can increase productivity by reducing the cell growth rate, which is basically less cells but more production. And also other antioxidants like glutathione, N-acetyl cysteine and ascorbic acid being studied, and they found that it reduces the ROS generation. And when studying the lysosomal activity, they found that when antioxidant being introduced, the protein aggregation has been decreased, and it also increases the thioredoxin reductase and peroxidase levels. And they found that the cell cycle inhibitors like CDKs are being increased in this case. Thereby the cell is being growth is being inhibited, whereas the production is being increased. Basically leads to productivity. Yeah. So basically the mitochondrial redox homeostasis is being restored. So these are the references.
please send it to my S-mail address, the link to your papers and your presentation. Both need to be sent. The meeting will also far they've been sent. Also, please give the S-mail address access to the papers and to the papers. You put in the presentation there. I've had access. It's just that the GK account has an access, so I have to use Wade and so on, just to make it useful. And yeah, please practice so that you can guess judgment.
Yeah.
Congratulations on finishing on time.
Yeah.
Okay, can you go to the light one?
Yeah.
Okay, so in the first point you have written in comparison to large scale, small scale superheated should
Yeah.
Why was that?
So as you know, large scale bioreactors will be made of stainless steel, and whereas the small scale will be made of glass. So basically, they found the light is being the small scale bioreactor is transparent to the light, whereas the large scale is not. So that's the reason why there isn't change in it. Of course, that's interesting because if I'm in that case, I won't think about light. I'll go for media composition or some other parameters. Yes.
What do you mean by productivity here? You told like cell growth will be decreased, but the productivity will increase.
So basically, productivity is the product formed per cell.
Which product is?
This is now monoclonal antibodies basically. So they found that if there is more product produced by a single cell, which means it's more robust and the productivity is high. Yes. Which one? Yes. Yeah. So this is an basically mRNA qPCR study. They found BCA-P is a protein chaperone, which is being increased in case of no antioxidant state, and DMOS is basically the solvent for this antioxidant. So in order to show that this DMOS doesn't have the influence on these results, they had a control which was supplemented only with DMSO, and they found that when antioxidant being increased, the level of these chaperone proteins has been maintained, and CHOP is a apoptotic protein which is being maintained at very low levels in case of an antioxidant presence. And same, they have done a Western blotting, so to show this. Yes. 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 Yeah. Yeah, basically, in antibodies was included, I think, in one of this thing. Yeah. So here, when this antioxidant being given, the fragmentation has been decreased, and that's because the oxidants has been neutralized by this particular antioxidant. Yeah. No, they haven't studied that. No. I think most of the people, they didn't check for the intracellular pH. And pH was not a consideration in any of this. Media pH, no, they didn't check the trends. They just checked for the viable cell density, viability, productivity, all these things. Maybe that could be checked, so could be one of